Hey, good morning. Thank you for the introduction. I'm Philipp Hanslovsky from the Saarfeld lab, specifically at the HHMI Junior Institute. And today I will introduce to you ImageLab, a Python package that bridges the chasm between two scientific image processing frameworks. On the one hand, Python with SciPy and NumPy. And on the other hand, we have Java with ImageJ and the image processing library ImageLab 2. When I first moved to the US from Heidelberg in Germany, I moved to the greater DC metropolitan area. I didn't know yet that I still had my greatest journey ahead of me, even though I just had crossed the Atlantic Ocean. The journey was not a spatial journey, though. It was a journey from one programming language to another, from one image processing framework to another. I first started programming about maybe a little less than 10 years ago, and during my master's in the Hambrecht lab in Heidelberg, and they use C++ and Python to produce high-performance software for image analysis, and they focus on neuron reconstruction from 3D data. If you're not familiar with neuron reconstruction, just think instant segmentation or cell tracking. Naturally, I was exposed to Python a lot, and after lots of work and lots of learning, I finally managed to understand the concepts, and I got very familiar with the SciPy frameworks, and I really started to like it. In this case, just to display a few, I show the Python logos, the SciPy logos, which you probably know by now, and the NumPy logo, which is the square with the yellow N on it, then the scikit image logo, with just the green and orange snake, and then matplotlib. These are just representative for myself. I bet everybody else in here in this room has their own favorite set of Python frameworks. I really like the concise syntax that you get with NumPy, the operator overloading, array arithmetics. So I was very happy on this side of the mountain, on this peak. And when I finally got to DC, I didn't even think that anybody would use anything else than NumPy for image processing. But I was very wrong about that, obviously. There's a whole other mountain, and I'm pretty sure there's many more, but so far I don't know only two. There's a whole other mountain, and this is the mountain that I will call ImageJ because it's like a collection of a lot of software. It took me a lot of time to climb this mountain. It was very hard work because I had to forget a lot of the concepts that I used during on the mountain SciPy, because they're not very efficient in Java or especially in ImageJ. But eventually, I reached the top of the mountain there, and just to show some software and frameworks there as well, I show the ImageJ2 logo, which is the microscope, then the ImageLab 2 image processing library on top, then the Fiji distribution, which is just Fiji, the blue letters, which ships a whole universe of scientific software and packages and modules that a lot of scientists use in their day-to-day research. This is particularly true for biomedical imaging, where I work now. And then there's BigDataViewer, which unfortunately does not have a logo, but this is my favorite tool in this whole environment. And it's a visualization tool for arbitrary large data, that volumetric data that reslices cross sections through volumetric data at arbitrary orientations and zoom levels. Other tools, like for example Mammut, with the icon of the mammoth there, built on top of BigDataViewer. Mammut is used for lineage tracking and visualization in cell tracking. And then I built my own tool on top of BigDataViewer, which is on the top right, Pantera, which is a tool for efficient generation of ground truth and proofreading in 3D connectomics or neuron reconstruction. How many in this room have ever heard of ImageJ or ever used ImageJ? That's more than I expected, but I guess if you're in this talk, then you know about that. But I still prepared something for those who do not know ImageJ or the world of ImageJ and think that image processing, you only need to do in NumPy, or there's only cool stuff in NumPy, the way that I thought about it a little more than 5 years ago. So I will demonstrate to you BigDataViewer in this little video, and I will show you how we can very freely and easily navigate this data set of this is an electron microscopy acquisition of a whole adult fly brain, and it is about 100 teravoxels. Just let that sink in. It's about 100 teravoxels, and we can freely navigate it without any lag or anything. And I hope that this will convince you that there's cool stuff on the Java side as well. So let's just play this video. So first, we can scroll through the data. We have an overlay, which you might hopefully see once we zoom in, see a little better, which is synapse predictions generated by Larissa Heinrich in our lab. And we can freely, arbitrarily orient the cross-sections. We can zoom in very far, so you get an impression of how vast the data actually is. We see the synapse prediction in places. We can change the contrast for each of the data sets individually, because those synapse predictions are not actually binary predictions, but more or less kind of probabilities. Not exactly, but that's just a detail. Then we can zoom out very far again, and then we turn off the raw data. And if you're familiar with NC82 stained light microscopy, then when we zoom out even more, this might look very familiar to you. And these are just the synapse predictions. We change the contrast a little, so we see them a little better. So all free navigation on the fly. And of course, we need to pre-process an image pyramid, but just to make this possible. But so you can see that there's really cool tools on the Java side. I hope I could convince you with this that it's definitely worth exploring this world if you haven't done that yet. All right, so let's go back to the slides. I've become very familiar with ImageJ and ImageLab 2 and all the frameworks on the Java side now. But still, there's so much cool stuff on this on the Python side, and all of you know that probably. So and it's just so hard to combine both frameworks in a single workflow because they don't really communicate to each other well. If you're familiar with wrapping C or C++ or other native code as Python code, then you might just tell me it's easy. You just need to create to expose your data structures as NumPy arrays, and then create wrappers for your functions and expose them to Python as well. Unfortunately, it's not as simple as that. To understand why that's not as simple for Java, we have to first have a look at Python. So what is Python? Python is an interpreted language with dynamic typing. That's why we love it so much. We don't have to write a lot of boilerplate code. We can just say image equals and then whatever NumPy array or something. We have access to native memory, which is super cool because then you can write very efficient software through C or C++ extensions. If you're not familiar with that, just think NumPy. You do like an addition of 2 arrays under the hood, very fast. C code guarantees that you get the most efficient array addition possible. And of course, there's this whole ecosystem of interactive shells and notebooks that scientists really, really love because they can document their code. They can just send those notebooks to someone else, and they're really easy to understand. Java, on the other hand, is a statically typed language that compiles into bytecode. And this bytecode is then executed in the Java virtual machine, also called just JVM for short. And this virtual machine adds an abstraction of memory so that any Java class doesn't really live in native memory, but in this abstraction layer that the JVM provides. And the Java the Java language API does not really provide an access to native memory. There's workarounds. For example, you could use the Java native interface to implement something in C and then provide that to your Java runtime. Or you can use internal methods of the Java runtime environment that are not part of the language specification. So hypothetically, they could be removed at any time, but it's been very stable, so that's good. But these options are usually a little more cumbersome, more work, and people who use Java, so they don't have to use C and don't have to worry about native memory. So in short, it's a problem of native memory versus JVM memory abstractions. And on top of that, even the difference between a Java array and a C array. So even though the names are the same and the syntax is kind of similar, they're definitely 2 very different objects. So the C array is essentially just a pointer into native memory, and the Java object is a Java array is a real Java object that lives in this abstracted memory layer. The ImageLab 2 library that I mentioned earlier is great because it separates or it virtualizes pixel access, which means any algorithm can see an ImageLab2 data structure and doesn't care at all where the data comes from. It could be a Java array, or it could be read from disk, or it could be streamed from the internet, or it could be entirely virtual. So, for example, a mathematical function that's evaluated at a pixel location. That also means if we have a way for Java to read native memory, we could also feed that into an ImageLab2 data structure. So there's basically two ways out of this dilemma of not being able to communicate between Java or ImageJ and NumPy. One of them is interprocess communication. That's pretty cool because it's fairly comparatively easy, and it can be extended to any language. Once you set up a protocol, you only need to implement it once per language instead of once per each pair of language, which is, of course, much less work. The downside is that we don't have shared memory, which is prohibitive for high-performance computing, like, for example, browsing through large amounts of data through BigDataViewer. If we have to send the data that we want to display in a Java view through a socket first, that's an unnecessary copy of data, and you even have to go through file I/O. So this will be a big slowdown. And we would prefer shared memory access. The other solution is to have Python and the JVM in the same process. And in that way, it's possible to have shared memory if we use the Java native interface and internal methods of the Java runtime to access native memory. And that way, we can avoid unnecessary copies of data. And for example, we can also modify NumPy arrays in place through Java methods. There's basically two options to achieve that. There's a Java implementation of Python, which is called Python. Which is pretty cool because you have access to all the everything that lives in the JVM, all the Java software, but you can still use Python syntax to code, which is a little more concise than Java syntax for those who know. And the downside is that Python implementation seems to have stalled at 2.7. And I hope everybody in this room is aware of what happens with Python 2 by the end of the year. So this is a big no. And Python 3 development, or Python 3 development seems to have stalled because there's not enough support in the community or industrial support. So unfortunately, that's not a way that we can go. Instead, I decided to start Java from within a Python process. In order for that to be possible, we first need to have a way to start the JVM within the Python process and then also expose methods and Java objects to the Python interpreter and user who wants to call them. I did not do that work. Luckily, this is already available in this package called PyGenius, which is a really clever name, as I would say, because it's JNI, the Java native interface, but also a genius. And this allows us to, from within a Python process, start the JVM, and it exposes classes that we import into the JVM, which is really all we need. On top of that, we need to have a layer that enables ImageLab2 and NumPy to communicate each other, so we can modify data without copying without copying. First, I created this ImageLab2 unsafe library, which uses internal methods of the Java runtime environment to expose native memory to ImageLab2, and now ImageLab2 data structures can read and write into native memory. That's the first step. Then as a second step, I created ImageLab2 ImageLab, which is just a convenience library that understands NumPy metadata, such as strides or offsets into native memory. Then finally I created the Python library ImageLab, which exposes all these other libraries into Python, and you can easily convert from a NumPy array into a ImageLab2 data structure. I want to stress again that this is only important because of this separation of concerns in ImageLab2, where you have a virtualization of pixel access that is entirely independent of how the data is provided. So we can just take the pointer of the NumPy array and feed it into an ImageLab2 image. And just to make sure that nobody confuses my contribution, I did not create PyGenius. That was already there, another open source library. So Yay for open source. And going back to my initial metaphor, looking at those two peaks, with ImageLab, I just created a small bridge that connects those two peaks, and now you don't have to walk down that mountain and climb up on the other side again. So I hope you all now want to know how to install ImageLab, and it's really easy. I added ImageLab and the dependency PyGenius to Conda Forge to make sure that it's always going to be up to date and available in the future. And all you need to do is run conda install for ImageLab. Then there's examples that you can install from the Python package index through pip. And there's more interactive notebooks available on my personal GitHub repository. If you don't copy this email right now, they will pop up at the end, and I will have the slides available online as well. So don't worry about not missing out on copying this link. How can we use it? So usage is pretty simple. We just need to import the ImageLab Python package, and then there's also a util package that we can use with some convenience methods. One thing that is very important is that you need to import ImageLab before you import anything from Genius, because internally ImageLab imports Genius and also adds some additional dependencies for the JVM. Genius starts the JVM once it is imported, and it can only have one JVM, and the JVM cannot modify it anymore after startup. So that's why you need to import ImageLab before Genius. Let's start with a very simple example. We want to simply wrap NumPy arrays as in ImageLib 2. So first we import ImageLab, and then of course we also want to import NumPy arrays. And let's see, yeah, okay, can you all see the mouse pointer here? Great. So we generate a random NumPy array of random numbers, and we just wrap it into an ImageLib 2 data structure. That's all that it takes already. So ImageLib 2 now has direct access to that memory of this NumPy array. And then we can use BigDataViewer to show this image. In a similar way, we create an ARGB image. It's probably a little different than what you're used to, because in NumPy world or SciPy world, ARGB images are usually channel images. And in ImageLib 2, ARGB images are scalar images with a 32-bit integer that maps into 4 8-bit channels. And we can use that special method, ImageLib, to ImageLib our ARGB to map to wrap in ARGB image. More complex examples are available in the ImageLib examples package that I linked earlier, and it provides a few command line commands that you can just run and see what happens, and then you can also look at the source code. I will show some more interactive notebooks, for examples, in a minute, but first I want to talk about known issues that we have at the moment. Java AW2 and Swing frameworks for graphical user interfaces don't play too well with macOS, unfortunately. So on macOS, if you use ImageLab and any software that runs on these frameworks, you have to use a wrapper script that is provided by ImageLab when you install it. But just to be aware of, if you run it on a Mac and it doesn't seem to work, that might be a good reason why. And for example, BigDataViewer that I will show in a bit uses a Swing, so you will have to run this wrapper script for that. Another current issue is that Dask arrays cannot be wrapped as ImageLib 2 data structures yet. So we cannot use, like for example, if you have a chain of Dask mappings, we cannot display that in BigDataViewer, which would be awesome, because then you could just put it at the end of your workflow and see what's happening right on the fly as it comes out. But unfortunately, that's not possible yet. But let's get back to the cool stuff and look at some examples. In the first example, I just want to show how you can open a NumPy array in BigDataViewer. And for a quick recap, BigDataViewer is a Java viewer for arbitrary large multi-view and 3D images and 3D image sequences. And it's developed by Tobias Peach, who is at the MPI CBG in Dresden. I have a link for the BigDataViewer and also a link for the notebook that I will show now. All those notebooks are also available online, and I will add the link at the last slide, so you can use them, try them yourself. So if you try them yourself. There's a little bit of text here that we don't really need right now. But first, we will import some Python packages, mostly h5py to load the data, because we're going to use a h5 file. Then there's a few other things that I import, because I have this convenience method to make sure that the data is actually there. And if it's not there, I will download it from Cremi, which is the biggest challenge for neuron reconstruction and synapse reconstruction that's currently available. So we run this cell. Then we import ImageLab, and we open the data. This will return a NumPy array, and then we just wrap the data. Running this cell takes a little longer because it has to start the JVM. There's a little lag there, and also just loading the data takes a short moment. But now we see the shape is 125 by 1250 by 1250, and then we just display a single slice of the data in the notebook, so we can confirm that we actually look at the image data. Let's do this. So this is a single slice displayed in the notebook, and now we want to open it in the BigDataViewer. And let's just run this cell. That's all you need to run, this ImageLab.util.bdv functions.show, and then you can pass a name. And there it is. We can navigate freely. What you will see here is that the data is anisotropic, which means that the resolution in X, Y, and Z is not the same. The resolution in X and Y is 4 nanometers, like one pixel in each in this, and in this direction is 4 nanometers. And 1 pixel in this direction is 40 nanometers. There's ways to adjust it in BigDataViewer. You can pass in a transformation that accounts for that, but it would just add some noise. So that's why this data looks a little shrunk along this dimension. So in case you don't believe me that it's actually shared data between NumPy and ImageLab 2, I have a little trick in hand here to try to convince you. So right now, I hope you can agree that we see some data here. And I hope that you can also agree that this will set everything in the array to 0. So we should have a black array. And if we run this, the data vanishes here. So this is just my little proof that it's actually a view onto the NumPy array.
Can you do a slice of it?
Can I do a slice of it? Yeah, I could. Let's just try this. But we have to reload the data again. Then I should I should comment this out because this is already running. Let's reload the data. Show it. And now let's make it a few slices, right? So let's do 30 to 90, something like this. I hope this is correct Python syntax. Yep, there it is. That's a good point. Maybe I'll just adapt the notebook to that.
Yeah, I think it's
more
confidence in it now.
Yeah. Okay, good. Great.
Why that might.
Okay, I'll definitely add that to the notebook. Thank you. Right. So the next example is also a BigDataViewer example again, but this time the data is just blank and there's nothing in it. But we will implement an extension for BigDataViewer in pure Python, and then we will be able to paint into the NumPy array with BigDataViewer. So we don't need this anymore, but we need this notebook. So we just have a few imports again, and then for now you don't need to be too concerned about what's happening here. I can explain it to you in detail, but that's not part of the session really. I just want you to know that this is all pure Python, and we're actually implementing a Java interface. And then finally, we create an empty image. I just add some color because I want to paint in color, so I don't want it to be black, so then we wouldn't see anything. Let me just show you the example because I'm running short on time. So here we have the example. It's 3D data again, and now we can just paint into the data. And this is actually writing into the NumPy array. So this is all pure Python. I had a few more examples, but I guess I'm running out of time, so I will just go back to the to the final slides. These are the resources that I used for the logos and the clip arts. And then the link for the slides is here. And there's more resources. There's a link to PyImageJ that you should check out if you're interested in ImageJ. And these are my social media accounts. Please contact me if you have any questions. I'll be here until Saturday morning. I'll leave probably in the afternoon. Thank you. So any questions? Yes, please.
Can you show us another demo?
Oh, yeah, absolutely. I have two more demos.
That, yeah.
I have two more demos if you're willing to sit through them. All right, there's. OK, so another demo is, let me show, because I have to acknowledge the people who make the software too. In my lab, John Bogovic, he created Bigwarp, which is a tool to manually align arbitrary data sets, really, by manually placing land points, and they can be arbitrarily large. He used it to align two different modalities, light and electron microscopy, of two different specimens of the same animal species. But we will just use it to warp a NumPy array. For example, here, Bigwarp. For this one, we will need some more Java dependencies that we can just import like this. And I will not explain what those cells do in too much detail, but now I will show you the software, Bigwarp. So we have one moving image that will change as we add points to it. And this will not change the fixed image. Usually these are two different modalities, for example, of microscopy, but we just want to warp this image. First, we need to add landmarks by just clicking. And whatever we click here, it will move somewhere here in this image, or here, and then this point will move here. And then once we have, I think, 4 pairs of landmarks, we can estimate a transformation, and this will be warped. And the cool thing is we can actually move this. Oh, no, that didn't work so well. We can, if I grab this right, we can move this image, and this is all virtual, so there no images are created. That's pretty cool. So now we have this Bigwarp thing, and we can grab the transformation parameters from it, and then we can use ImageLab 2 to use this transformation to actually warp a NumPy array. And there we have a NumPy array, and this is actually the NumPy array that we warped. And now if we change the warping again, something like this, a little bit crazy. So this is probably a very bad setting of landmark pairs. Then we can get the parameters again and update the array. If you want one more example, I can show that too. If you're sure.
Yeah, we have time.
And then the last example that I have is my own software, Pantera. That's a tool for proofreading, neuron segmentation, and for generating dense ground truth. And it has one thing that's pretty cool, is that you can visualize any label data in 3D without pre-computing meshes. So we can visualize any NumPy array that we want 3D. So let's look at this real quick. So first we create some fake data, just periodic data that's for the raw data, and then the labels are just connected components that are thresholded on the raw data. And then we just load it in Pantera. There it is. And because this is BigDataViewer based, we can also reslice any way we want. And once we select a connected component or an object in here, it will render it in 3D without pre-computing any meshes. And this is just a plain NumPy array that we use here. And that's just another example. All right, I think now I'm out of examples. If you have any specific questions to these examples, just approach me. I'll be here. Contact me on social media. I'll try to get on the Slack, but I'm not on there yet. But yeah, just approach me. Thank you for listening.
Thank you.